Okay, so hi, my name is Iman Syada binti Nur Azmi, and my group will be presenting about the HIV, which is known as human immunodeficiency virus. So my part, I will be presenting about the history of HIV. So from many research, scientists have managed to trace the origin of HIV, and they have found that HIV may be come from the chimpanzees and the simian immunodeficiency virus, which is also known as SIV. So SIV, they are actually a HIV-like virus, but they only attack the immune system of the apes and chimpanzees. So, in 1999, researchers has identified a strain of chimpanzee SIV, which is called as SIV-CPZ. And this SIV-CPZ is nearly identical to the HIV. So, the chimps, the scientists found later, they actually hunt and eat two smaller species of monkeys, which is red-capped mangabeys and the greater spot-nosed monkeys. So, these chimps, they will actually carry two strains of SIV, which is likely to combine to form SIV-CPZ, the strains of chimpanzee SIV, which the researchers have found. These two strains likely combine and spread between chimpanzees and humans, and this virus can infect the humans when they enter the human body, which can happen when the humans hunt and eat the chimpanzees, or when the virus enter to the body through an open wound. So, the first transmission found of SIV to HIV in humans can be found in the global pandemic that occurred in 1920, which is in Kinshasa. That is actually the capital and the largest city in the Democratic Republic of Congo. So that's all from me. Thank you.
Hi, okay, now I will explain about the morphology of the HIV virus. HIV virus is roughly spherical with a diameter of about 1 to 0 nanometer. HIV virus has a very high genetic variability. It is composed of two copies of positive strain RNA which held together by protein P71. HIV virus is enclosed by a conical capsid composed of viral protein P24. This is the one that is capsid protein P24, the blue one. Then the RNA genome consists of 9 genes, which is 3 of these genes are GAG, pool, and envelope. Okay, this gene contain information needed to make the structural protein for the new virus particle. Inside the capsid protein P21, there are 3 enzymes required for HIV replication, which is reverse transcriptase, protease, and integrase. Reverse transcriptase, protease, and integrase. As for matrix, it is composed of viral protein P17. This is the matrix of the protein of the RNA, the yellow one. The matrix protein P71 is surround the capsid. It is to ensure the integrity of virion particle. Matrix, the yellow one, is also surrounded by a phospholipid bilayer, which is embedded by 70 copies of glycoprotein composed of GP120 and transmembrane glycoprotein GP41, which is um, this one, the blue one, and the red, the stick light red one. This structure has 70 copies. So that's all from me about morphology of HIV virus. That's all. Thank you.
Assalamualaikum and hi everyone. I want to talk about the test for HIV infection. So, HIV tests are detect the presence of the human immunodeficiency virus (HIV), the virus that causes acquired immunodeficiency syndrome (AIDS) in serum or our blood, saliva, and urine. So, the test may detect the antibodies and antigen or RNA. So, the test that usually people do is the antibody test, which our previous semester we have been run this ELISA test, which is enzyme link 7 assay. It is very high sensitivity towards this. So first step to do this antibody test. First, the person's serum or blood diluted to 400 fold and applied to a plate to which HIV antigen has been attached to it first. And then, if the antibodies to HIV are present in the serum or in the blood, then they may bind to this HIV antigen. So the plate then washed to remove all other components of the serum, and then the secondary antibody that binds to the human antibody then applied to that plate and followed by another wash. And then this antibody is chemically linked in advance to the enzyme. So the plate will contain enzyme in proportion to the amount of the secondary antibody binds to the plate. So the subscript for the enzyme applied and change the color to fluorescent if it positive. So next is antigen test, which is the P24 antigen test detect the presence of the P24 protein of the HIV, which is the capsid protein of the virus. So the monoclonal antibodies specific to the P24 protein are mixed with the person's blood. These three molecules mix together, and the NEP24 protein in the person's blood will stick to the monoclonal antibody and an enzyme linked antibody. So the monoclonal antibody to P24 cause the color change if the P24 was present in the sample, which is HIV positive person blood. So P24 antigen tests are used for early detection only for HIV, as antigen P24 rises soon after infection relative to antibody. So the presence of P24 antigen diminishes as the body increases production of antibody to the P24 protein, making the P24 more difficult to detect later. So another test was conducted to be more effective for these purposes, which is nucleic acid testing. So for the third is antigen antibody combination test. So this test designed to detect both P24 antigen and HIV antibody in a single test. So it can detect as early as 2 to 6 weeks after infection, and it's highly recommended in laboratory testing. So next is the NAT, which is nucleic acid based test. So this test detect one or more targeted sequence located specifically to HIV genes, which is HIV gag, HIV envelope and HIV polymerase. But it is very expensive to run this test. So because it is using the polymerase chain reaction PCR, using reverse transcriptase that convert the RNA viral gene to the cDNA. So last but not least, one other test that used in the HIV treatment is CD4 T cell count. So CD4 T cell count is not a viral HIV test, but it is between the CD4 test in the blood in the HIV positive person. So it considered to be a marker to the progression of the HIV virus. So for the normal CD4, for the normal person, the CD4 is 500 to 1000 cell per microliter. But for the HIV positive persons, which contain AIDS, it will drop below 200 cell per microliter. So in general, the lower T cell, the lower immune system functions. So this test is good to measure the immune system of the HIV positive person, to them taking the right portion of medicine to prevent it to become AIDS more seriously. So the next is the 10 early signs and symptoms of HIV/AIDS. HIV infection is one of the serious and life-threatening health concerns worldwide. Consequently, many people lost their life if they don't do the diagnosis and treat the disease promptly. So there has three stages of HIV virus, which is acute HIV infection, which is the flu-like symptom that occur within first 2 to 4 weeks of the contracting HIV infection. Next is chronic HIV infection, which is known as clinical latency, which is the chronic HIV infection stage can last for decades. So last is AIDS. It occurs when the CD4 cell count below 200 cell per microliter and vulnerable to opportunistic infection. So the different stage of HIV infection determine the risk of infecting other people and our life as well if we have the HIV positive blood. So the 10 early warning signs of HIV infection is mouth ulcer, muscle and joint pain, a sore throat, swollen lymph nodes, night sweating, diarrhea, rashes, fever, growing tired and fatigue for a long time, and headaches for a long period from the usual. So the symptoms usually show in early 2 weeks after the exposure and can be within 2 months late for the symptom to appear. So there is no cure, but we have the prevention from the virus growing into AIDS. So cure can be in the term of prevention and taking the medicine for the HIV positive person for his or her lifetime. So that's all for me. Thank you.
Hello everyone, so today I'm going to share on the stages of the HIV. So HIV has 3 stages, which is the first one is the acute infection, second is the clinical latency, or we call as the chronic HIV infection, and lastly is AIDS. So during these stages, the large amounts of virus are being produced in our body. So generally develop within 2 or 4 weeks after a person had been infected with the HIV. And during this time, some people might have flu-like symptoms, for example, like the headache, fever, and rash. So during this time, HIV starts to multiply rapidly and spread throughout the body, and the virus will start to attack and destroy the infection fighting, which is CD4 cells of the immune system. And during these acute infections, the level of the HIV in blood is very high, which generally increase the risk of the HIV transmission. Okay, second, we have the clinical latency, also called as the chronic HIV infection. So during this stage, HIV will continue rapidly but at a very low levels, and it is still active. And people with a chronic HIV infection may not have the HIV-related symptom, but they can spread the HIV to the others. And without the treatment, without the treatments of the HIV with the HIV medicines or vaccines, the chronic HIV infection usually advances to the AIDS in like 10 years or longer than that. Okay, for the last stage is AIDS, which is the most severe stages. Okay, during this stage, HIV has severely damaged the immune system, so the body cannot fight off the opportunistic pathogen. And for example, the opportunistic infection like the infection related to the cancer, and during this time, uh, the opportunistic pathogens start to attack and damage our cell in the body. So the people that diagnosed with the AIDS, they have uh, they have 200 cells millimeter square in the body, which is the CD4 cell. So without this treatment, people will survive, typically will survive 3 years or less than that. Okay, as you can see, this, okay, I would like to share you this picture. So this is a CD4 cell that have been put in the test tube. You can see that before the HIV infection, you would have lots of the CD4. But during the first stages, which is the acute HIV infection, your CD4 start to decrease, and it will be decreased more during these stages, which is the chronic HIV infection, and lastly, the last stages, which is the AIDS. And also, okay, this is one of the pictures that I would like to share. So as you can see here, during the first stages, the CD4 concentration will be higher and it start to decrease at the second stage, and it will be more lower during the last stage, which is the 8th stage. So you can see during the 8th stage, the CD4 concentration will be less than 200 cells per milliliter square. So this is the HIV concentration and the antibody concentration. So you can see during the 8th stage, the HIV concentration will be higher than the antibody concentration. Okay, I think that's.
Hi, my name is Aisha. Okay, now I will explain to you about how people can get infected with HIV. As you can see in this picture, these are how HIV can be transmitted. First, sex without a condom, or we can call it unprotected sex. So HIV patient need to wear condom to prevent from transmission to their partner, because HIV can be transmitted by fluid transmission or blood contact. During having sexual intercourse may be resulting in tear or abrasion, so that will allow the fluid transmission or blood contact to their partner. Next one. Okay, now I will explain to you about my next presentation, how to treat HIV. How people of HIV can get treatment. First, of chemotherapy associated with cancer, because usually HIV patient is very high risk of getting cancer. Because why? Because HIV patients are very weak, have weakened immune system and immunosuppression and inflammation that could lead to the development of cancer. So HIV patient have high risk of getting cancer. Chemotherapy increasing number of drugs at least temporarily control reproduction of virus. But the obstacle to treat HIV as developing a vaccine is high mutation rate of the HIV virus quickly lead to resistant strain and the persistence of latent viral reservoir. Okay, as you can see, Kaposi sarcoma, non-Hodgkin lymphoma, invasive cervical cancer defines that person is infected with HIV and AIDS. Means if these people getting Kaposi sarcoma and all these cancer, it confirms that these people will have the HIV as well as AIDS. Okay. Next one is fusion and cell entry inhibitor. This inhibitor, this drug will block the fusion of HIV virus with the cell and prevent the viral entry to the cell. As we learned before, how HIV can enter the host cell is via two things, which is receptor and co-receptor. Receptor is CD4 receptor on the immune cells, CD4 T cells. So GP120 on envelope of HIV virus must bind to receptor CD4, and then once accomplished, it must bind GP120 of virus must bind to the co-receptor CXCR5 or CXCR4. Once accomplished, the binding, the virus, once accomplished, the GP41 will expose. Means conformational changes occur. When GP41 expose and the conformational changes occur, the cell of virus and it will bring the virus cell and membrane cell together and fusion occur, and then virion can be released into the cell. So this drug will bind to the GP41. Then the conformational changes cannot happen, and thus the fusion or cell entry of virus cannot occur. For example, drug enforvirtide target the GP41 region of viral envelope, which facilitate fusion of the virus. Next one is reverse transcriptase inhibitor, or we call it antiretroviral drugs. Nucleoside reverse transcriptase inhibitor will terminate the viral DNA by competitive inhibition analog as nucleoside, because this drug will be as analog nucleoside. Analog nucleoside will compete with natural nucleoside and continue and bind to the chain. Because of analog nucleoside is different structure from the natural nucleoside, when it bind to continue the chain of DNA, it would not form chemical bond, so reverse transcription cannot happen. This is the way that drug inhibitor will prevent the reverse transcription, and it also disrupt the synthesis of new piece of proviral DNA. Next, last one is protease inhibitor. Protease inhibitor is to block the activity of protease enzyme, which HIV virus really need protease enzyme to cleave the polyprotein of HIV to smaller protein, so that it is very important for assembly of the viral HIV protein. Even though HIV can still replicate in the presence of protease inhibitor, the resulting variant still immature and cannot infect the new cell.
Is about the replication or the life cycle of HIV in human body. Okay, the first step is by, as you can see in this picture, as stated, number one is the attachment of the virus on human cell. This is called is binding or the fusions of HIV. This HIV begin its life when they are bind to the receptor called CD4 and CD4 and CD5. Okay, when they start to attach, the second step is the reverse transcription. As we know that HIV is very special because it have a reverse transcriptase enzyme that can make them to replicate in our body. The second step, this HIV enzyme called reverse transcriptase at these stages will convert the single strand of HIV to double-stranded HIV DNA. And when it starts to reverse transcriptase, they will go further to the third stage, which is integration. Integration means this virus will DNA. Sorry, the newly formed of HIV DNA will enter the nucleus of the host, and it start to integrate. And this integrated HIV DNA is called virus. This virus may remain inactive for several years and producing no copies. Some will further the process and some will just remain in the nucleus. And after they enter and the release the cell will go as transcription, when the host cell receive a signal to become active. There's now some active and some remain. When this host receive the signal, the single strands become active, and this provirus use a host enzyme called RNA polymerase to create copies of HIV genetic material. Okay. And as well as a shorter strand or RNA messenger, mRNA. This mRNA are used as a blueprint to make long chain of HIV. Then to make a long chain of HIV protein. Then the fifth step is assembled. The HIV enzyme called protease will cut this long chain HIV protein into a smaller individual protein, and this smaller HIV protein come together with copies of HIV RNA genetic material, a new virus particle assembled together, and they will start to budding. When they start to budding, and it will during the budding, the newly assembled virus push out and it will form bud, and during the budding the virus still the part of the outer envelope, and when it's ready to bud, it will bud out from the cell. Okay, that's the 2T that I want to share to all of you. Okay, I think that's all for me. Thank you.
Hi friends. Okay, today I'm going to share with you about the challenges in developing HIV vaccines. Okay, there are many challenges in developing HIV vaccines. Okay, I want to focus in few, which is the antibodies can have different properties. So-called binding antibodies simply attach to part of HIV and may or may not have antiviral effects. Okay, the next one is HIV continually evolves because of genetic mutation and recombination. Okay, once HIV infection becomes established in one body, however, the virus continually undergoes changes and many variants may arise within an infected person. So the scientists having difficulties to produce or make an HIV vaccine due to these factors. Okay, there are many factors in affecting HIV progression in our body. Some of that is age. When the age increases, the progression of AIDS will be much slower. Besides, with increasing age, there are low levels of chemokines to interfere with HIV's ability to infect CD4 T cells. The next factor is infection route. Through blood transfusion and drug injection causes high clinical progression and mortality of diseases. So, through blood transfusion and drug injection, the HIV progression will be higher. The next is pregnancy. Viral load increases in the 12 weeks after giving birth, even in women on stable antiretroviral therapy, and that viral load gradually increases thereafter. Thank you.